Okay, everyone, this will be the last video, I think, for the last video related to bacterial conjugation. And if you watched any of the previous videos in this series, the Lecture 9 series, I mentioned my hands are pretty grody here from working on, they're doing some plumbing at my house yesterday. I don't know that much about plumbing, which is probably why I wasn't wearing gloves. But hopefully next week my hands will be clean and it won't be so embarrassing to make these videos for my students. Okay, so in this video we are going to talk about an experiment called interrupted mating. I feel like I'm spelling this wrong. There should be like 2 R's or 2 P's. Two R's, interrupted mating. So an interrupted mating experiment allows us to determine the order of genes on a bacterial chromosome. And it was the first type of experiment that allowed scientists to do or achieve such a feat. So how does this work? Let me take you through an interrupted mating experiment, and by going through this example you should be able to answer, I think, the couple of example problems we have assigned for this part of the course. So if we have 2 bacterial cells here, one cell is an HFR cell for high frequency of recombination. I described how these cells are made in the last video. And let's assume this is an F- cell, and the genotypes of these cells are threonine plus, Lu plus, Lac plus, Gal plus, and streptomycin sensitive. Now, what does this mean? Okay, this means that this gene for making the amino acid threonine is functional, wild type. Leucine for making the amino acid leucine is functional. A gene for being able to utilize lactose as a carbon source is functional. Same thing for this. A gene for being able to utilize galactose is functional. And strep S, so this, what this means is that this gene right here is sensitive to the antibiotic streptomycin. So if we try to grow this HFR strain on media containing the antibiotic streptomycin, it's not going to grow because the bacterium is not resistant to that antibiotic. So the F- strain, the genotypes for that, 3-, Lu-, Lac-, Gal-, and Strep R. So this bacterium here can resist the antibiotic streptomycin, but it has mutated copies of all the genes needed to make threonine, needed to make leucine, needed to utilize lactose, needed to utilize galactose. So now we can use these 2 strains in an interrupted mating experiment to determine the order of these genes on a bacterial chromosome, or on the bacterial chromosome. So let's say this is the chromosome of the bacterium. There's one over here. And let's say right here is the integration of the F factor. And when we cut the DNA here during conjugation, the single strand is going to be unwound and delivered through the conjugation tube into the F- string. Okay, so let's say we do this experiment here. We mix these 2 cells together, make a giant cell suspension, and we give the cells not much time at all to conjugate. So DNA starts going through, it doesn't even make it through, let's say. Let's call this the 0-minute time point. We essentially give it no time for conjugation. So this is the HFR strain, this is the F- strain, And we have threonine here. So we do a bunch of, once we do this, what we will do is the first thing we should ask ourselves, okay, we mix these cells, we give them 0 minutes to actually conjugate and for the HFR strain to deliver DNA into the F- cell. We give no time to the HFR strains to conjugate and deliver DNA into F- cells. This isn't just one interaction between a pair of cells. We're looking at interactions between thousands of cells in a suspension containing millions of different individuals of 2 genotypes. So we take a sample at 0 minutes. So the sample, none of the cells in the sample had time to actually conjugate. And how are we going to get rid of the HFR cells, right, to do our media test? Because we want to see, of the F- cells, how many of them have this gene now, 3+, how many have LU+, how many have LAC+, and how many have GAL+. And we can do this with a series of media-based assays. We can determine, of the F- cells, how many are still 3-, how many are LU-, things like that. We can determine how, well, I should rephrase that. We can determine how many of these types of cells got a functional copy of 3+, LU+, LAC+, or GAL+. So, but we have to get rid of the HFR cells. So to do that, what we do is we just, plate the cells on media containing streptomycin, right? Because all the F- cells can resist the antibiotic streptomycin. Okay, so that's going to be in all of our media. And through a various number of assays, we can determine whether the F- cells are now 3+, LU+, LAC+, or GAL+. And let's say at 0 minutes, none of these cells, none of them are 3+, none of the F- cells are LU+, none are LAC+, none are GAL+. Well, we could take another sample at 5 minutes. So 5 minutes after mixing these cells, or hundreds of millions of each type of cell in a cell suspension. And let's say here's the chromosome in each one. And now, let's say, okay, a good chunk of the chromosome has made it through at that point. And let's say, we do the media test again. We get rid of all of the HFR cells in the sample by plating on streptomycin antibiotic containing plates. And we're able to see that, okay, we covered 5 colonies or 5 cells that are now LU+. So F- cells used to be LU-, now they're LU+. So what that suggests is that of these 4 genes, LU+ was the first one to enter the F- cell. And none of the others must have entered at this 5-minute time point. So we can start making our map of the gene order on the bacterial chromosome, and the map is relative to the fertility factor. So the fertility factor is first, and LU+ must come next because it was the first one that was delivered into the F- cell. So we can check at 15 minutes. We'll take a sample at 15 minutes. And at 15 minutes, a lot more of the chromosome, that single-stranded DNA from the chromosome, has been inserted into the F- cell. So now at this point, we might have 25 LU+, and now let's say we see some cells that no longer require threonine in the media showing up. So 25 no longer require leucine in the medium, 5 no longer require threonine. So what does that mean? Okay, so that means threonine, what's 0, there's still 0 that none of that, we don't recover any that are LAC+, we don't recover any that are GAL+. So that suggests threonine must come next. So look, all right, we've discovered the order of the Lu genes and the threonine genes on the bacterial chromosome by doing this interrupted mating experiment. Okay, so let's take a look at 30 minutes. And you can probably see where I'm going with this. Another one of these genes is probably going to have time to make it through into the cell before we interrupt the mating. And look, a lot of the chromosome has made it in, and here is the original chromosome over here. So, okay, we interrupt the mating here at 30 minutes. Now let's see, we have, say, 100 of these and 63 of these and 20 of these, but still 0 of these. Okay, well, that would suggest that gal plus comes next. And if we should look at 60 minutes or an hour and a half or something, well, then maybe that's probably when lac plus would come in. So, okay, so we've used this interrupted mating experiment to determine that Lu comes before threonine, comes before galactose, which comes before lactose, or the genes, what should I say? The Lu gene comes before the threonine gene, comes before the galactose gene, which comes before the lactose gene. I don't know that if this is really the order of these genes in E. coli. This is just a hypothetical example of how an interrupted mating experiment would work. And I should say, how do we interrupt the mating? We take a sample, but then you can blend the suspension of bacteria in a blender, and that is supposed to be sufficient enough for breaking the conjugation tube and ending the mating interaction between these two cells, or groups of two cells. And I should also say mating, bacterial mating is another word for bacterial conjugation. So that is how an interrupted mating experiment can work. And what you should remember is that interrupted mating experiments can be used to determine the order of genes on a bacterial chromosome. And that was how things were done in, I think, the 50s and 60s. I think maybe the first interrupted mating experiment was in the 60s, I believe. So today, you know, we can do genome sequencing and things like that. So we have much more efficient processes for determining the order of genes on a chromosome. But by going over this experiment, we can learn how in nature bacteria can transfer genetic information from one to another. And we learn a little bit more about the conjugation process and how genes are delivered in a linear manner during the interrupted mating experiment, well, during conjugation. Okay, well, I hope that helps you understand how interrupted mating works. And please take a look at the problems. And again, as always, if you have any questions, email me, set up extra office hour appointments, or come to office hours, or ask some questions in the lecture.